All right, in this video, we actually have the process of transcription, all right, going on. That's the technical term for how RNA polymerase copies the DNA and makes an mRNA as a result that you're going to eventually send out into the cytoplasm. So this, of course, occurs in the nucleus where the DNA is stored, the chromosome, and there will be, of course, a small piece of DNA that we have identified as a gene, and that gene will be copied. All right, now here we have an animation. As you can see, we have the parts that I mentioned earlier, the things that make something a gene. What makes something a gene? Well, it's a series of letters of chemicals. We represent the chemicals adenine, cytosine, thymine, and guanine as the letters A, G, C, and T. And these chemicals make up DNA, and as you can see, you have double-stranded DNA. So this is indicating that this DNA is part of an organism, a chromosome, something like that, which it is that's the way I have it described here. And we have two strands that bind together. The A's bind to the T's and the G's bind to the C's, etc. All right. Now we have one strand that's called the reading strand and one strand that we're going to call the sense strand, and I'll explain why they're called that in just a second. Or let's do it now. The reading strand is the one that's actually read. So if you'll see here, the red ball here is RNA polymerase, which is the copy machine in Grandma's cookbook. So it's a protein that sticks to the DNA, and it sticks to, as we said, to the promoter. And one of the many promoters are TATA, T-A-T-A. So I've put T-A-T here, and I've highlighted it as the promoter. And so when RNA polymerase gets near the DNA, it is chemically attracted to the promoter. It is chemically attracted to this TATA sequence, and it sticks. And when it does, it's going to move across this strand of DNA, and it's going to make a copy. And that's why we call this the reading strand. This is the strand that has the TATA on it that attracts the polymerase, all right, and this is the strand that's going to be read. Well, when it makes a copy, it doesn't make an identical copy. It makes the copy, all right, that is over here on this side. It makes an mRNA copy of this, but it's called the complement, all right. This strand over here is the complement of the top strand. So the bottom is the complement, we say, of the top, and the top is the complement of the bottom. They are complementary. And what, of course, that means is whenever you have an A here, you have a T here, and whenever you have a G here, you have a C here, etc. That's what complementarity means. Well, when you make the copy, you don't make an identical copy of this sequence. Instead, you make a complementary copy. In other words, the little sequence here that's being made, this is the mRNA, the little green sequence in this little animation. Well, that little green sequence that's being created as the RNA polymerase moves across the gene, it looks a lot like this. The difference, of course, is that it's making an mRNA copy. So RNA in green here, all right? That green, notice there are U's in it and not T's. And that's the difference between DNA. So these two strands, these are DNA. The green strand here, all right, that's being copied from this one, it's a complementary copy, if you want to say it that way. Well, that is mRNA, and you're going to replace it, the T's, with U's. Now, if you'll notice, if this is a complement, that means it looks like this strand down here. That's why we call this the sense strand, because the sense strand is the one that's identical to the mRNA. So if you really want to just know what the mRNA of your DNA is, you just go look down here and you go, okay, here's the TATA. It's going to be ATAT, all right, and then it's going to start here. So what is our mRNA going to look like? Well, it's going to look like this region right here, all right, in between the promoter and the terminator. That's the region it's going to look like. And all we're going to do is replace the T's with U's. So if you have a chance to pause this video, all right, and you look at this green sequence, you should see that it is identical to this sequence down here, in between here and here. It's identical, all right, with the exception of wherever there's a T, you're going to put a U, because this is RNA. Now, this RNA that is created from our gene, this is a gene, all right, the copy itself is called an mRNA, or a messenger RNA. And this is what will be leaving the nucleus, all right? This is the copy, all right, in Grandma's cookbook. It will leave the nucleus and it will go to the cytoplasm to be read by a ribosome. The ribosome will read this little strand here, this little green sequence, all right, and it will translate it. It has the ability to understand what this says, and what this says is how to build a protein, and that's going to be for another video.